So these are, for me, really the big challenges in mouse genetics, indeed for the next century, because I suspect we'll still be working on this in 100 years' time. We want to characterize the function of every gene in the mammalian genome. And both yesterday and today, you'll hear more talks about generating mutations in every gene in the mouse genome. But for all of those mutations that we're generating, we need to characterize the phenotype of all those mutant mice. And it's that work, generating mutations, characterizing the phenotype of those mutant mice, that is going to give us a more systematic and comprehensive view of the function of all of the genes in the mouse genome. And of course, a byproduct of this, a very important byproduct, is identifying models of human disease. Now, there are enormous challenges to phenotyping and understanding the relationship between genes and phenotypes in both mouse and human. And it's a sobering thought that, in fact, until the major project started recently that you're going to hear more about today, around 2/3 of the genes in the mouse genome remain to be knocked out and investigated. We know still bizarrely little about the function of most of the genes in the mouse and human genomes. And for the knockouts that have already been generated, for about 30% of the genes in the mouse genome, those that have been studied, the legacy data that we have in the databases is very patchy and incomplete. And that partly reflects the fact that the phenotype investigations that have already gone on outside in the community, by and large, reflect the interests and the expertise of the investigator that's actually looking at those knockouts. So an investigator makes a mutant in a mouse. They might think that that mutant is important in kidney function. So they look at the kidneys and they document what happens to the kidneys in that particular mutant. But of course, they don't necessarily look at the vestibular system. They don't necessarily look at the heart. They don't ask, what is that gene doing systematically across all organ and physiological systems? So we need to move in phenotyping in the mouse to a broad-based phenotyping approach, a systematic and comprehensive phenotyping approach, and an approach that is applied to all of the genes in the mouse genome. Now, one of the advantages of that approach is that it's entirely unbiased. We don't make any assumptions about what gene is functioning for what reason, in what tissue, in what organ system, in what physiological system. So it's a very powerful way for generating hypotheses about what genes might be doing in an unbiased way. So these are the major challenges that face us in mouse genetics as we move forward over the coming couple of decades. And what I want to do over the next couple of talks is flesh out how we address those challenges, what's happened so far in terms of understanding how we undertake broad-based phenotyping of knockouts or other mutations for every gene in the mouse genome, what kind of data will be produced, and what impact will that have on biology in general. And I think it's fair to say that we in the mouse genetics community believe that the projects that are now ongoing in mouse phenotyping will be truly transformative for biology in general. So this is really what I'm going to focus on. As we go from genome to function to disease model, the two talks that I'm giving today are going to focus on what have we done to develop broad-based systematic phenotyping methods for the mouse, and what are we doing in the future to phenotype a mutant for every gene in the mouse genome. So the challenges of phenotyping. I think they can be summarized by looking at this matrix, because effectively this matrix summarizes what we need to gather in terms of all of the data about the relationship between gene and function. And it's a three-dimensional matrix. On this matrix, we have millions of data points that reflect, first of all, the mutant that we're looking at, the genetic background that mutant's on, and I'll come back to that, the particular tests that we're applying to that mutant. We have a whole series of phenotyping tests that effectively could go up to infinity. And of course, along another axis, and I'll come back to this later on, we have environment. So our aim is to populate this matrix with data points systematically and comprehensively. And when this matrix is full, we'll really have a proper and complete understanding of the relationship between gene and function. So of course, if we go back in time to look at the kind of data that's been produced and is currently in the databases, if you go to the MGI database at JAX, what we find is that the matrix is populated in a very patchy fashion. There are specific data points where an investigator has applied a particular test to a particular mutant under a particular environmental condition. But what we want to do is fill in this matrix almost completely. We want to be able to complete the data set, and through completing the data sets, we'll really be able to analyze the genetic systems in a much more profound way than we have previously. We have phenotype discovery, characterizing disease models through this multi-systems approach that I've just described. We need to do multi-systems phenotyping, broad-based phenotyping, where every mutant mice that we take is thoroughly investigated for all of its organ, physiological, biochemical systems, also cellular and molecular systems. Because without those broad-based screens, we won't understand systems genetics. We won't really understand the interplay between individual genes and function and the interplay between the genes themselves. But of course, if we're going to develop these broad-based screens, if you think about it, we need to identify efficient primary, if you like, multi-system first-pass indicators of interesting disease models. We've got to think carefully about the tests that we apply. Because if we go back to that matrix, clearly we can have many, many tests. But there's going to be a cost implication, as I'll come on to. We have to think about how best do we screen our mice? Do we provide this primary broad-based screen, which will ultimately lead us to not only identifying the phenotypes of those mice, the most important phenotypes, but also the interesting disease models as well. And this leads me into what are some of the critical issues, and you'll see these critical issues played out in everything that I say today. There are several critical issues that we need to address. The first of these I call the testing conundrum, and there are a number of issues here. Of course, in the mouse, we've got about 20,000 genes. There's a lot of genes in the mouse and human genomes, and that means that we're going to have to make a lot of mutants, and we're going to have to look at a lot of mice. So depending upon the number of mice that we test through all of our phenotype tests, maybe it's an N of 7 or an N of 10, there's going to be a large number of mice that we're going to need to test. We also need to think that when we carry out a phenotype test, how good is it at measuring a difference between a wild-type control mouse and a mutant mouse? This we call test variability. Depending upon the variability of the test, how robust the test is, that will to some extent determine the effect size that we can measure. And that effect size is also determined by the number of mice that we test. Also, of course, the effect size that we can detect has got to be clinically relevant. We have to be able to identify differences between our mutant and control mice that have some relevance to clinical disease models, to phenotypes that are going to be interesting to the wider disease community. So you can see from all of this that there's a calculation to be done about the number of mice that you test for each gene, each mutation that you create. And as you'll see, generally we think we have to analyze between 7 and 10 mice to enable us for most of the tests that we carry out to detect a reasonably clinically relevant effect size. So if you just think about it, 20,000 genes, 10 mice, 10 males, 10 females for each gene, that's an awful lot of mice that we have to test. And of course, that's just for one allele for each gene. If we want to do multiple alleles, or for example, if we want to do alleles on different genetic backgrounds that might affect the phenotypic outcome. So immediately you can see phenotyping begins to expand and expand and expand. The other critical issues are standardization. Clearly, if we're going to phenotype mutations for every gene in the mouse genome, this is something that can't be done at one center. It has to be a multinational effort, and I'll come back to this later on. But it also means that we need to standardize what we're doing in phenotyping. We need phenotyping SOPs, we need to standardize housing conditions, diets, I'll come back to this later on. And also we need the informatics. We need to be able to collect all of this phenotyping information, which is produced in a standardized way, into some central database that you and others can look at to find out what was the phenotype of my particular mutant mouse. So let me just elaborate on some of those points. So our aims, in a sense, in terms of mouse phenotyping, are to develop robust, broad-based comprehensive phenotyping platforms able to deliver phenotypic information for all body systems. But in doing so, we've got to develop standardized phenotyping protocols that work well and will detect the effects that we want to see in our phenotypic systems. So we've got to standardize our protocols so that we can share and compare phenotype data from mouse genetic centers throughout the world. And of course, we've got to ensure the associated informatics developments, protocol descriptions, data exchange standards, ontologies, how we describe the phenotypes that you're going to hear more about later on. I can't stress too much in terms of phenotyping the importance of standardization. We need better reproducibility of test outcome, both across time and across laboratories, where we need better comparability of test outcome that allows us to share phenome results and bring them together in a unified database. And this is perhaps the biggest challenge for mouse phenotyping. Our vision is centers around the world taking different mutants, phenotyping them with standardized phenotyping protocols, and then bringing that data together in a unified database. But clearly, we've got to all be doing phenotyping in the same way so that we have some comparability about the data and the data standards. So let me describe how we've begun to address this through, in fact, a European model. It started here in Italy and other countries around Europe, of trying to address this issue of, can we actually work together as a multinational consortium to take large numbers of mutants and phenotype them in a standardized way, bring them all together in a database, and disseminate that information out to the wider world. And this is the model that I'm going to tell you about in the first part of my talk. And it relates to some of the critical European mouse programs that have developed over the last 10 years or so. I'm not going to tell you about all of these programs, but just to say that you'll hear more about the EUCOM program, I think later on today from Wolfgang Wurst. But this program was, in fact, and is still underway, is developing mouse mutants for most of the genes in the mouse genome. So this is the bread and butter of what we do. It's producing the mutants that we're then going to phenotype. And indeed, just like our phenotyping, it's producing them in a standardized way. I'll tell you, I'll allude a bit to that, but I'm not going to tell you a lot about it because you'll hear more about it later on. But the two programs that are really critical, I'm going to tell you more about, are the Eumorphia program, which is now done and dusted, but it was to develop and standardize the mouse phenotyping platforms that we need to analyze all of our mutants. And then the EUMODIC program, which is the one I'm really going to focus on, which is a major pilot program to utilize those standardized phenotyping platforms to analyze all of the mutants that are coming out of the EUCOM program. So let me just allude a bit to Eumorphia, which happened in the early part of the last decade, where a consortium of laboratories around Europe got together and said, we have to have a set of standardized phenotyping assays. We need to make sure that those tests work well and that they are consistent across different laboratories. And this was the Eumorphia program. So, in fact, if you go back to an issue of Nature Genetics back in 2005, you'll see that we created this database called EMPRESS, the European Mouse Phenotyping Resource for Standardized Screens, that effectively is a database of validated standard operating procedures for phenotyping a mouse. And you can go to this URL and you can actually see all of these standardized procedures. And in fact, these standardized procedures are now being updated as we move from a European program of multinational phenotyping to an international program. In my second talk, I'm going to tell you more about that international program, but for the moment, I'm going to focus on the European program. So Eumorphia got all of these institutions around Europe, got together, and they worked hard at both devising standardized operating procedures and then testing them to see, did they really give consistent results across all of these mouse centers? And the answer, by and large, was yes. So here's a particular example of ensuring that a particular test gave us reliability, robustness, and reproducibility. So what I've summarized on this slide here is, in fact, the results of an open field test. This is measuring the movement of a mouse in an open field. And this is an important neurobehavioral measure. It, in fact, measures things like anxiety of the mouse, because the time spent in the center of the arena is a measure of the level of anxiety of the mouse. So the more anxious a mouse is, the more that it will keep to the edges of the arena. So this is an important neurobehavioral test. And this just is one of the parameters that's measured from the open field test. This is the amount of time, the percentage time that's spent in the center of the arena for a particular mouse. And here are the results from 4 different institutions: CNR here in Italy, the GSF in Germany, MRC Harwell in the UK, and ICS in France. And what they've done is they've looked at measures of percentage center time for 4 different inbred strains: C57 Black 6, C3H, Bulb C, and 129. And the important thing you can see is that although the absolute values vary somewhat between the different institutions, which is not a surprise, the important thing is that the ranking of the different strains is reproduced between the different centers. And this is absolutely critical. It shows that the genetic differences between these strains that make some more anxious and some less anxious can be detected in a robust and reproducible way between the different centers. So this is a validated test. This is a good SOP that's working between different centers, and we can now utilize this for phenotyping our mice. Now here's an example of a test that when we started out with our SOPs, it's all over the place. And you can immediately see it's not working. There's no consistency in the strain ranking across the 5 centers here. This is the rotarod test. And here we're measuring, so you put the mice on this spinning wheel and you ask, how long can they stay on this spinning wheel? This is the measure of latency. When they drop off, that's the time that they've managed to spend on the wheel. So it's a measure of motor coordination. The longer the time they can spend on the rotating wheel, the better coordinated they are. So it's a bit like a balance test. Different inbred strains, we know, will show different performances. Some are better motor coordinated than others. But you can see that when we measure this across the different inbred strains, the data is all over the place. So we went back, we looked at the SOP, we asked questions about why it wasn't consistent, why it wasn't working. I won't go into the details. But we came up with a modification for the test, which involved altering particularly the cover on the rod. We changed that. And we actually shortened and simplified the test somewhat. And as you can see in this second round of validation, we then began to produce data that was much more consistent. So this is the kind of work that we need to do to ensure that we're utilizing phenotype assays where we can share and compare data across many institutions around the world, which will be absolutely critical for creating this unified database where we fill in all of the points across this matrix, where we have particular mutants, we have a particular genetic background, we have a particular environment, and we carry out all of these standardized, robust, and reproducible tests to fill in this matrix of data. So that's where Eumorphia got to. We have some ideas about how to proceed with broad-based phenotyping on large numbers of genes. So let's see, can we actually do it? Can we take hundreds of mutant genes, provide a broad-based phenotyping for each of those mutants, and begin to collect and analyze the data? And this was really the role of the EUMODIC program, which is just completed. I'm going to tell you quite a bit about that because it's given us enormous insight into how to do large-scale phenotyping, which is now being translated into an international effort, which is going to be the focus of my second talk today. So I'm not going to tell you much more about it, but because you're going to hear more about this as well later today. But the International Knockout Mouse Consortium, of which EUCOM is a major part, I've already mentioned EUCOM to you, has been making knockouts in embryonic stem cell lines for 5 or more years. And in fact, this slide is a bit out of date now. It's more like 15,000. They now have around 15,000 knockout embryonic stem cell lines, which can be converted into knockout mutant mice. I must stress, wait for later on. I won't go into the detail today, but you're going to hear more about these particular mutants. These mutants are all standardized. They're produced by a single targeting vector. And they produce an allele which is called knockout first conditional ready. This is the allele here, TM1A. And this is the allele that was, in fact, analyzed in the EUMODIC program. So all of the mutants have this particular structure. Now, please don't get worried that I haven't gone into the complexities of this particular allele at the moment. You're going to hear more about this later, and we'll discuss it more. But suffice to say that we're utilizing a standardized knockout allele. So we're looking at the knockout mutations, and all of these alleles were produced on one single standardized genetic background, black 6N. All of the ES cells are black 6N. So all of these knockout embryonic stem cells are being made into mouse in the EUMODIC program, and these are the ones that are being phenotyped. We'll talk more about this later on, but suffice to say, these are the alleles that we're using in the EUMODIC program, and in fact, we're using them in this program that I'm going to tell you about later, the International Mouse Phenotyping Consortium as well. But suffice to say, at the moment, we're using a standardized knockout allele on a standardized genetic background, black 6N. So EUMODIC was set up a number of years ago, 5 or 6 years ago, to undertake the first major pilot program to utilize a standardized phenotyping pipeline, which I'll tell you about in a moment, called EMPRESSslim. And the idea was to apply this pipeline to the analysis of 500 of these mouse mutants. Standardized phenotyping on a relatively large number of mutants, capture all of the data, and see what it provides us. This work was carried out across 4 major mouse centers in Europe. And the broad aims of this program were to look at all of the issues of logistics, operational issues, phenotyping issues, informatics issues, in terms of a large-scale, broad-based primary phenotyping of hundreds of knockouts. So we looked at logistics of mouse production and phenotyping, the utility of the assays, the sensitivity of the assays, and also, as I've alluded to, the informatics issues in order to capture this information that's coming from multiple centers and get it out to the wider community. And this is the effective workflow of the program. So we're taking, as I've described, these alleles from the EUCOM program, the mouse mutagenesis program. We're making them into mice. We're breeding heterozygous mutants. We interbreed those to produce homozygotes. And where the homozygotes are viable, they go into a primary phenotyping pipeline called EMPRESSslim. 7 males and 7 females go through this primary phenotyping pipeline. And this is being carried out at these 4 mouse clinics in Germany, France, UK. And all of the data that comes out of that primary phenotyping pipeline goes into the Europhenome database. Now, of course, in some cases, our homozygote mutants are not going to be viable. And when that happened, we returned to the heterozygotes, and we phenotyped the heterozygotes. This is the primary phenotyping pipeline that we used in EUMODIC, EMPRESSslim. And indeed, in fact, it's two pipelines, pipeline 1 and pipeline 2. And overall, there are 20 phenotyping platforms covering a number of different biological and disease areas, as you can see. So pipeline 1 goes through dysmorphology, blood pressure, calorimetry, and so on. And here's pipeline 2, which has a variety of neurobehavioral and sensory tests, followed by some hematology and clinical chemistry and facts analysis. Although it says 10 males and 10 females on this slide, generally we put at least 7 males and 7 females through each of these pipelines. So you can see that this is an enormous amount of work just of itself. 500 mutants, generating the pedigrees, the cohorts of mice, mutant mice, to go through each of these pipelines. The pipeline runs from 9 weeks when it starts to 16 weeks of age. Now, as I said, there are 20 phenotyping platforms here, and they measure over 400 different phenotype parameters, because in each test you measure a number of different parameters. And also we collect a large slew of metadata as well. There are over 140 metadata parameters. So we're collecting information on diet, environment, the equipment that was used, the actual technician that did the test, and so on and so forth. So there's an enormous amount of collateral data that reflects the environment that might impact upon the outcome of the test as well. So this program's now complete. So let me tell you a bit about the data that's come out of it. So 500 lines were completed, and the data, the phenotype data that was captured from going through the EMPRESSslim phenotyping pipeline has now been entered into Eurofenome. We've QC'd the data for nearly 460 lines, and we've analyzed those. So this is the data that I'm going to show you now. I should say that all of these mutant lines are available through the European Mouse Mutant Archive. And we finished the data analysis last year, and this is the data that I'm going to show you before. But if you're interested to actually look at the data, you can go straight to the Eurofenome database, go to www.eurofenome.org, and you have a variety of tools to look at the data and to actually see what's there, either across multiple genes or for individual genes or for a particular test. There's an enormous number of different tools that allow you to dissect and analyze the data in different ways. But the take-home point is that, and this I think just shows you the overall ambition of the program, is that this involved the analysis of over 30,000 mice. There's nearly 10 million data points in this database, and there's over 3,000 phenotype annotations. And this is just on 500 mutant lines. So you've got to think of how we would scale that to go from 500 genes to 20,000 genes. As I said, you can go in and look at the data. Perhaps one of the best ways to look at the data is to look at these heat maps. These heat maps, effectively in the red, indicate where you've got highly significant phenotypic hits, where the mutants are significantly different from the controls. So in fact, you can select individual genes and look at the heat maps and look at whether, either in males or females for particular tests, you actually see a significant phenotypic difference. So to take an example here, here's this transmembrane protein. In terms of male body weight, it's got a significant difference from the control. But I stress, just go into the database and play around with it. It's very easy. So generally, what did we find in terms of identifying phenotypes for all of these lines? So I'm going to give you some general data from EUMODIC, from the analysis of 458 lines. And these are phenotypes which had a very high significance level. So these are the ones that we think are robust phenotypic differences. So here's a chart of the number of lines with that many annotations. Here are the number of phenotype annotations going from mice that had no phenotype annotations through to, well, here's a single mouse here which in fact had 88 phenotype annotations. You can see that 84% of the lines have an annotated phenotype. But the most important thing for me is that nearly 75% of the lines have more than one annotation. Now this was very, very important. So of course one of the critical things that we want to find is that we don't expect genes to be just working in one organ and one physiological system. We expect them to be doing multiple functions. This is known as pleiotropy, and pleiotropy is being revealed. The fact that we see many lines with multiple phenotype annotations was very satisfying to us. So we have a high hit rate in terms of identifying annotations, phenotype annotations, and many of the lines show multiple annotations, as we would expect. If we include phenotypes for viability and fertility, nearly 90% of the lines have a phenotype annotation. And it's also important to note that a very large number of the lines were homozygous lethal. Around a third of the lines are homozygous lethal. Over 10% are subviable. They show some lethality in the homozygous state. There are a few lines with reduced lifespan, and around 50% of the lines are viable as well. Let's look at the difference between homozygous mutations and heterozygous mutations. There is a little small difference that more homozygotes seem to be annotated with phenotypes than heterozygotes. And there's a slightly higher mean annotation count for the homozygotes than for heterozygotes, but there's not a huge difference. Heterozygotes have almost as many interesting phenotype annotations as the homozygotes. But around half of the genes that we looked at in EUMODIC had no data on them whatsoever. This just shows the big gap there is in our knowledge about gene function. So well over 200 lines had nothing in the database about what those genes did. And what we found was that for 85% of those, 180 of those, we were able to add significant phenotypic annotations. And this allows us to search diverse systems for novel disease genes. And let me just show you some examples of that. So I think it's quite important. So in fact, we've gone to 3 biological areas: diabetes and obesity, bone, and neurobehavior. And in each of those areas, you can't read this slide very well, but we've chosen tests that have parameters that impact upon those disease or biological areas. Each of these circles represents one of those parameters. So in these Venn diagrams, we've identified genes which have hits in those different parameters. And we might expect that genes with combinations of hits in those particular parameters are giving us novel insights into diabetes and obesity. These are novel genes that haven't been annotated before that are giving us phenotypic hits that suggest that they have some involvement in either diabetes, obesity, bone, or neurobehavior. And you can see that there are many genes giving us many different hits that are suggestive of their involvement in those particular disease or biological areas. So let me just bring out some examples for you, since the slide's rather difficult to read. So here's a gene called MARC2. Never been annotated before. We know nothing about it, but it's got various hits in the diabetes and obesity parameters that suggest that it's involved in that particular disease or biological area. And it's interesting that there's a related gene, MARC1, which came up in a GWAS study for reduced total and LDL cholesterol. So a novel gene that suggests that we ought to consider this as a candidate in diabetes and obesity pathways. Here's another one in bone. This glycosyltransferase, never been studied before, so we've made a mutant. We've looked at the parameter hits within bone. We see that it's giving us some important changes that are suggestive that this gene indicates a novel role in glycosylation of proteins involved in bone formation. I could go on and on, but here's one from neurobehavior. The GBX1 gene, a gastrulation brain homeobox1 gene, has an anxiety phenotype, suggesting that it has a role in specifying populations of cholinergic and GABAergic neurons, but never been looked at before in the context of adult phenotypes. But of course, we find genes such as this gene, BRAE, again, never been looked at before. It's known to be, through protein studies, a component of the BRCA1A complex. But as you can see, it has a wide range of functions. We find it affecting parameters in the neurobehavioral area, the bone area, the diabetes and obesity area. Genes with pleiotropic functions, giving us the first insight into what these particular genes do. If we look at classifying the number of annotations we have across the different biological areas, the different ontology terms that indicate our different biological areas, all the way from behavior through to reproductive systems, you can see that we're capturing information in different systems to different extents. So I think an important thing going forward, and I'll reflect again on this in my second talk, is that, of course, these phenotyping pipelines are as good as they get. And to some extent, of course, phenotyping is how long is a piece of string. You can always do more phenotyping. And we'll always be in the situation that we won't necessarily be recovering information in all of our systems in the most maximal and efficient way. And there's certainly, from the EMPRESSslim pipeline in EUMODIC, there are some areas where we think we're very deficient in picking up phenotypes that we'd be very interested in, particularly disease phenotypes. So in fact, the number of annotations that we pick up in the cardiovascular system is rather poor. So this indicates that the pipeline needs improving. We'll always need to improve our phenotyping tests and our pipeline to deliver the insight that we need into different disease and biological pathways. So I want to finish with this particular talk in actually, and it's something that I'll come back to some extent in the second talk, about saying that, clearly in EUMODIC we've demonstrated that we can do large-scale phenotyping. We can get a lot of very interesting and novel phenotyping information. But what are we not doing? And Roger yesterday talked about the aging ENU screen at Harwell. Clearly, what we're not doing in the EUMODIC phenotyping pipeline, where we look at phenotypes between 9 and 16 weeks of age, we're not looking at aging. Indeed, we're not doing all sorts of challenges that we might do to reveal additional phenotypes. And I just want to make this point, bring this point home to you. So, of course, aging, through the aging ENU screen, which Roger talked about yesterday, is an important component of phenotyping, but one that's very difficult to deliver across large scale. So if we want to do hundreds of mutants and we want to age them out to 1 year, that's an enormous amount of space and cost that would be needed to identify those particular phenotypes. Of course, as Roger alluded to yesterday, we're doing an ENU aging screen at Harwell. We're not yet providing or applying aging as a challenge to the EUMODIC program or future programs on mouse knockouts. But the need to identify genetic networks involved in age-related disease, of course, is absolutely critical, because most of the important diseases in the human population are diseases of aging, as I'm sure Roger emphasized yesterday. So we need to be able to identify those genetic networks, and we need to think of ways of being able to capture phenotypes on all these mutants we're generating and aging them as well. And that's a difficult thing. Of course, in the ENU aging screen, we have a screen, a pipeline that captures much of the information that was in EMPRESSslim, and I'm sure Roger showed you this yesterday. So we do have the tools, and we do have the experience to age our mutant mice and to put them again through these pipelines to capture additional phenotypes that would be revealed by the challenge of aging. And I'm sure we would find many additional phenotypes, because already, and this is just to remind you of what Roger told you about yesterday, in the ENU aging screen, we're getting close to 150 pedigrees entering the aging screen, and some half of those are showing age-related phenotypes. So I think this gives us a clear indication that if we were to do the same thing for all of the knockout mutants that are being generated in EUMODIC or other programs, we would find a lot of additional phenotypes. And we'd find phenotypes, I'm sure, in that 10% of mice for which we're not identifying any phenotypes so far. So aging is a critical challenge that we need to be able to apply to all phenotyping pipelines. But how we do that, the best way to do that, and how we get the money to do it, that's another matter. But aging isn't the only potential challenge that we need to think about in mouse phenotyping. We can think of other challenges. We can think of diet, high-fat diets, for example. We can stress our mice by giving them a so-called McDonald's diet. We can think about doing our screens under different microbial status, different background levels of pathogens and microbes in our mice. We can do our screens where the mice are carrying particular infections, or we can just use infection as a challenge to identify genes involved in host resistance. We can do our phenotyping under physical stress to see what additional phenotypes are manifested. We can think of the social environment for the mice, how we house the mice. And we can think of even things that you wouldn't think are terribly important, but are certainly important to me. One of my big interests is the genetics of deafness. We can house our mice where they're stressed by additional noise. Of course, that's a big problem in the human population. We're all exposed to increasing amounts of noise, which leads to deafness and age-related hearing loss. So we can use these to identify additional phenotypes. And I just go back to our age-related screen at Harwell, where we have an age-related screen for hearing loss or presbycusis. Here's one of the mutants, which is revealed at 9 months, wasn't present at 3 months. So this is an age-related hearing loss mutant. It would be interesting to apply pulses of high frequency, high decibel noise to the mice and see whether we can elicit some of these additional phenotypes by stressing the mice with additional exposure to noise. So overall, there's a huge area of work that we need to think about exploring of these additional challenges which might sensitize our mice to reveal additional phenotypes that we wouldn't find just by the most straightforward screen. So I come back to this matrix again. This is our big challenge, where we're developing tests, robust pipelines that will reveal the phenotypes for particular mutants on particular genetic backgrounds. And this, of course, is what I've just stressed here, the environment. We need to both control the environment, but we also need to change the environment to elicit all of the phenotypes and all of the genetic functions that lie within the mouse and human genome. So in this first lecture, I hope I've given you an idea of the principles that are surrounding large-scale mouse phenotyping, its utility for exploring gene function and revealing novel disease models. We really are beginning to address these challenges now, and in my second lecture, I'm going to say something about how we're moving forward in the future and what we're doing there on a wider international basis over the next 10 years to actually phenotype mutants for every gene in the mouse genome. An enormous but challenging project. But it only remains for me to thank all of the people in the EUMODIC program. These are all of the people, pretty much, who worked in EUMODIC over many years across the 4 centers. And I, of course, alluded to the aging program at Harwell. Which these were all of the people involved in Harwell in the ENU aging screen. And importantly, this is what we're going to come on to next after the coffee break. Moving from what I've talked about here to the challenge of developing an encyclopedia of mammalian gene function and talking about the International Mouse Phenotyping Consortium. So I'll leave it there. Thank you for your attention, and I'll take questions. Thank you.